So as Lauren said, I am a postdoctoral researcher, and I'm at Arizona State University in working with Jeffrey Jensen, and I'm a postdoc there. So yeah, I want to talk to you. We work in population genetics. Population genetics is sort of a study of genetic variation across individuals in a population. And by being able to study this kind of genetic variation in populations, by, let's say, sequencing DNA from different individuals in the human population, let's say, we can arrive at things like what happened in the past, how did humans migrate, what were their movements like, what were the migration rates, and historical bottlenecks that were experienced by these populations. And this sort of work can, of course, inform us about how diversity in the world today is shaped. More importantly, population genetics can also, for instance, help us understand evolution of pathogenic species within hosts. So it's relevant, of course, to evolution of SARS-CoV-2 in human populations. So by being able to, for example, model the changes in allele frequency of these different mutations that occur in pathogenic species, we can arrive at predictions for instance, resistant mutations, etc., in these type of populations. But really, to be able to model these kinds of populations accurately and make such inferences, we really need to understand how the different evolutionary forces that act in a population, how they shape the genetic variation in the population. So for instance, here, these colorful circles are showing you they're just a representative of genetic variation in a population. And this variation can change over time. If you take a snapshot of the population at any point in time, you can try to understand what type of evolutionary forces act on it. Usually the evolutionary processes that we talk about are selection, mutation, recombination, and sort of changes in population size. For the purpose of this talk, it's not important to know how exactly these processes together govern these patterns of variation. But what I do want to emphasize here is that although we can mathematically write down expressions for, say, individually one or two or even three of these evolutionary forces acting together in a population, it's been a very difficult task to do that for all of these evolutionary processes together and sort of modeling realistic natural populations evolving. So what do I really mean by that? So on the left-hand side here, I'm showing you sort of a complex population history of our natural history for population. So, when populations out in the nature, they don't usually, they can undergo fluctuations in population size, for instance. So here I'm showing you three different colors. The yellow, red, and blue, they represent different populations. So natural populations can split from each other, occupy different areas, have migration in between them, and can also undergo individual changes in population size. And so modeling this kind of complexity is quite difficult, actually, only simply theoretically, for instance. And on top of this, the added complication on top of this is that when we think about genetic variation, such variation does not exist independently. All organisms have chromosomes, and so a lot of mutations are linked to each other on these large chromosomes. And this sort of linkage determines how the trajectory of these mutations would be in the population. And so modeling the sort of complex genome architecture where genes are sort of non-uniformly distributed across the genome, that sort of adds another complexity or layer to these populations. And so when we really want to understand the dynamics of mutations in natural populations, it ends up requiring simulations, at least at this point. And so one of the ways that we can make inferences from these sort of natural populations, for instance, is by using sort of like these Bayesian methods, which we call approximate Bayesian computation. So for instance, if you want to make inferences about two parameters, theta and gamma, from a natural population, what one would do is that you'd have a prior on these parameters, and you sample from this prior, sort of a wide prior that you don't know much about. You sample from these priors and you perform lots and lots of simulations. And then these simulations can be used to obtain some sort of a summary statistic or like a statistic to measure the state of the population. And then these observations can be compared to the observed data. And using this sort of a matching sort of technique, one can sort of generate the most likely posterior distribution of both of these parameters. And so this is sort of a very popular technique that's now being used to be able to make inferences from DNA sequence variation data in natural populations. And I just want to emphasize that when we do these sort of simulations, we require a lot of these simulations. And so here I'm showing you only 4 parameters, alpha, beta, gamma, delta. But really these sort of models start from like 4 or 5 parameters and can go up to about 30 or so. So these are parameter rich models. And so, you simulate each of these parameter combinations. Each parameter combination is usually simulated with multiple replicates going from about 1 to 1,000, could even be 10,000. And of course, this number, that is the total number of parameter combinations that we simulate, that can really end up being a large number sometimes, starting from like a minimum of 3,000 to almost 1 million such parameter combinations. And so if we want to do a quick calculation here, if each of these simulations was about 10 minutes, we were simulating 1,000 reps and we were doing 3,000 combinations, it would require 500,000 hours. And this is a pretty large number for especially, sort of regular clusters, for example, provided by universities. It would be a very difficult thing to complete so many simulations there. And so that's where the OSG comes in and the HTCondor system comes in. I was really looking at one point to try to understand how I could manage to complete so many simulations and how this would be possible. And that's when Gil Spire, who works at Arizona State University, he's part of the research computing staff there. He happened to find me and it encouraged me and introduced me actually to OSG, and encouraged me to join that and think about that, and gave me enough information that I could set up my appointment with Lauren Michael, who then set up, gave me a good introduction and told me how I could use OSG to do this project. So I'd like to talk a little bit first about like the resources that I found helpful while working with OSG. And so of course we all know the sort of the quick start page, which I think is very, very helpful and useful for OSG, where all of the different kinds of help exists on the same page and you can easily navigate it. I also found Christina Koch's tutorial for introduction to OSG very, very helpful. So I think there is a PDF of this online available. I have given the link to that down there, and that was very helpful to sort of give an overview of the HTCondor system and also, just to understand the structure of how things work on HTCondor. This tutorial is also very useful for sort of how to modify submit files, etc., where I think she gives quite a bit of detailed information about submit files and how you can change how you can make variables in the files, etc. So I think that was very helpful. I've also pointed here to 2 other links that are useful for generating submit files in general, or modifying them for your own usage. One of them is, of course, the OSG support page, and another page here. So next, I want to talk to you about some important and helpful HTCondor features and OSG support structures that I found helpful for particularly my project. So, I want to tell you a little bit about how we sort of run these type of simulations, or what's really involved here. And the first thing, I wanted to say is that these kinds of high throughput simulations, they are made very easy on HTCondor. I mean, I think this is basically the right setup to perform such kind of work. So, so what we really try to do, for example, is first we have some type of population parameters that we are modeling. That is parameters that describe the entire population. So in this case, I'm just sort of saying, for example, population sizes that describe the population over time, let's say. And so some combination of these parameters will describe that particular population. And I call this sort of a simulation number. So if, for instance, we need 3,000 parameter combinations of these population parameters, there would be, for example, simulation 1 to simulation 3,000 in this set. And then for each of these population parameters, we simulate or sample sort of genes across the genome. So these could be about 100 genes, or they could be 1,000 genes. It depends on your particular analysis and what you want. But each of these genes that you sample from the genome would have a specific length, for instance, would have a particular rate of mutation occurring there, a particular rate of recombination. And so these are all parameters that are now sort of varying across this genome that we are sampling. And for each of these population parameters, we have these multiple genes that we're simulating. So which could be about 100 or 1,000 or so. And this I called a gene number. So you'd have a gene number that goes from 1 to 100 or 1,000. And then for each of these scenarios, right, we have replicates for each of these. And so each particular gene in a particular population parameter would have about either 100 replicates or 1,000 replicates, again, depending on what the scale of your study is, or how accurate you want your results to be, versus how much time you want to spend simulating these. And this would be called a replicate number. And so in my case, this is sort of like a pretty nice and sort of a straightforward way for me to go ahead and set up my submit files. So what I do in my submit files is that I have my argument line, which which has basically the simulation number, the gene number, the replicate number, the random seed, and then all of these different parameters that are associated with each of these: the simulation number, gene number, the recombination rate, the length, etc. And so I have these arguments set up that I just queue from a file, and it's a file that is specific to that simulation number. So let's say simulation 478. It's a .csv file that looks something like this, where for that file, you just have all of these parameters, and these numbers sort of correspond to these parameters written up here. And so this actually becomes quite easy. I only need to transfer a .csv file and my script file for performing these simulations. And just, by just being able to submit one of these simulation numbers, I can at the same time basically simulate all my gene numbers and rep numbers for that particular simulation number. So this is really convenient. It's very easy to submit these sort of files or perform these simulations in OSG. The second thing that I found very useful is OSG allowing accessibility to containers. This is, of course, just a support page that's listed for OSG, and I use a container for basically the one software that I use, which is Slim. Slim is this really great sort of simulator for population genetic data, where it doesn't really require any input files or anything. It just requires parameters as arguments, and it performs the simulations and sends out an output file. And so it's a really convenient sort of simulator for doing these things. And it also uses a single core. And so I can just use one simulation for, every one core can just use, can perform one of these simulations. So I also want to specifically thank Christina Koch here, because she helped make this container for me when I was starting off. And it's been really great because I've just been able to use it. I think this is a pretty great feature. And I feel like, if there can be more of these sort of containers that can be accessible, that could really help sort of at least this kind of bioinformatics or evolutionary genomics type of research, where you can then just simply sort of almost like adding a module right into your work. You can just go ahead and use all of these programs, which I found very interesting. And third, I just wanted to point out that the data storage at OSG has been really fantastic for me. This is, of course, from the web page of OSG, and just pointing out how much data we all have available. And I thought I find that very useful, because all of these outputs of simulations that I get, I do need to store them somewhere. And it's been great to have this much storage space to keep them there, especially because if sometimes we realize during our simulations that we've made an error, or we need to update our pipelines in some manner, and we need to rerun them. And it's useful to be able to keep this sort of a backup there at the data storage part. So I think that's great. So what I wanted to show you quickly here is that what we have been doing is we've been doing this for different species. So Drosophila, I just want to show you that each of these replicate on an average takes about 20 minutes. And we performed 1,000 replicates of each with 3,000 parameter combinations, and that's about 1 million hours would be required based on these numbers. Of course, it's not exactly this, because this is just a mean number here. We also performed a project with influenza genomes, which took much longer, about 3 hours. And so we also used much fewer number of replicates and parameter combinations, but that also was quite a few number of hours. And we are now working with human populations here that take, and this is sort of work in progress right now, where we intend to complete this project as well. We have really been able to get a lot of work done in this scenario, with about 9.66 million wall hours that I could get. This is all actually simulations of Drosophila, and humans is still in progress right now, and will hopefully reach the same level as the Drosophila level in a few months. And we've also published 3 manuscripts by now, where OSG was definitely a key computing resource. Without OSG, it would not have been possible to complete any of this work. So that has been really great. There are 2 papers for Drosophila, one for the influenza and human cytomegalovirus study, and we're still working on one for humans and primates. So I just want to end with the importance of OSG in the future for population genetics. So I want to emphasize again that we don't yet have mathematical expressions describing patterns of genetic variation in populations that involve complex population history and selection acting on these linked genes on a chromosome. And simulations are a great way to understand these sort of complex evolutionary scenarios that we cannot yet mathematically model. So and we know that simulation frameworks in population genetics are becoming faster and much more efficient. And so I'm really hoping that OSG can play a really central role in performing these sort of evolutionary inferences in the future. So with that, I'd like to thank you for listening. And I also want to acknowledge all of the people who have helped set up the OSG resource and my advisor, Jeffrey Jensen. Thank you so much, and I can take questions.
All right, thank you, Parul. Any questions can go in the chat, or again, using the raise hand option in your reactions menu on Zoom. And I believe Parul will be joining us in the discussion session right after this too. I'll also add Gil Spire, we've been in touch with several times. You mentioned him, he's staff at ASU, has really pointed researchers to OSG Connect. There are a handful of projects active in the last year and last several years. And for those of you joining us tomorrow, there will be a presentation from ASU's Director of Research Computing in our set of campus presentations. So ASU will be presenting the full story here between Parul's work and tomorrow's perspective from Doug Genowine. Parul says, sorry about the cat. I thought the cat was a great addition. Somebody's asking if you can introduce the cat.
Right, now the cat seems to have disappeared and it's not coming up here.
All right, well, thank you again.
Thank you.
I was also looking at that link you provided to a page that explains submit files, and it has some really neat explanations of some of the submit file aspects that I think we'll have to check out and maybe think about incorporating into some of our documentation too.
That sounds great.
Yeah, and the video, by the way, that Christina developed is actually like authoritative Condor tutorial for users of Condor throughout the world and everything, but it certainly applies to just about any Condor system, including the Open Science Pool. So,
okay.
Yeah, our facilitators often wear multiple hats, and those of us at UW tend to help with Condor things here and there.
Nice.
All right. Well, thank you again, Parul. We'll look forward to seeing you in the discussion session where people can ask you some more questions.